I'm Dr. Suzita. I'm from Biomedical Science Department, and I'm the moderator for the second talk. Our talk today will be by Dr. Soon Hao. He is a senior lecturer from our Biomedical Science Department. He's our expert in neurovirology, specifically focusing on the pathogenesis of neurotropic viruses, such as enterovirus A71, Japanese encephalitis virus, and also Nipah virus. His research bridges the gap between basic virology and clinical neuropathology, and he utilizes advanced mouse models to understand how viruses invade the central nervous system. And I'll pass the floor to Dr. Tan now so that he can tell us more. So thank you, Dr. Tan.
Thank you. Thank you, Susie. Hi, good morning, everyone. So this is actually a paper that published in previous year. It's not about the clinical work, but it's more towards the basic science. So let's go into the thing. The background is actually about EV-A71’s. So this is a virus that causes the hand-foot-mouth disease. But sometimes, in very rare cases, it can actually escalate into the CNS and cause a severe and fatal neurological complications. So these are the outcome: aesthetic meningitis, poliomyelitis, light paralysis, and the encephalomyelitis. So what we know about the CNS infections from the autopsy, basically across the patients, it shows a serotype pathology. It's an inflammation in the spinal cord, specific region in the brainstem, hypothalamus, and the dentate nucleus. So there are lots of ongoing research trying to understand how EV-71 actually enter the CNS by bloods crossing the blood-brain barrier, or utilize the peripheral or retrograde axonal transports along the motor nerve. Because in the autopsy patients, we only often see this CNS is infected, but not the other organs except the skin. We do not know how this virus actually transports into the CNS. There are more and more evidence shows that it can go into the CNS by serous way, but the most major discussions over time would be the retrograde axonal transport along the motor nerve. Right. This paper specifically answers certain questions. Number one, how does the virus spread within the neurons? So once it gets into the CNS, how it spreads, why there are certain types of the patterns inside the autopsy brains, and is the SCARB2, which is the viral entry receptor for this virus, play an important role in the spreadings of the virus within the brain. So to answer this, we would like to investigate how the EV-71 disseminate after the IC or intracerebral inoculations of the virus into the brain directly. And this is our hypothesis, right? The retrograde still play an important role. It will flow back to the virus will flow back to the blood and causes a viremia, and it will utilize the retrograde axonal transport back into the brain itself, rather than intra or interneuronal spread within the CNS. Secondly, the viral entry receptor SCARB2, here we're using the mouse model, so it's a mouse SCARB2M SCARB2, is not responsible or is independent towards the inflammation patterns that we observe in the brain region. So these are our mouse model, right? Using a 2-week-old mice, we inoculate them with the EV-A71 mouse adapted strains, analyze using the histopath analysis and some viral molecular analysis, for example, the titrations and molecular technique. Then we also have some in vitro results to counter the analysis of the animal model. To answer that, we select 2 regions in the brain. One is the thalamus. The second one is the pons medulla. So in human autopsy cases and in our mouse model cases, you often observe that the pons medulla is heavily affected or heavily infected by the CNS, by the virus. in the CNS, whereas the thalamus or hypothalamus is often spare or less severe affected in both humans and mouse model. So after inoculations, we will keep them for observations throughout the days. We notice that all inoculate mice from both groups, hypothalamus and the pons infected groups, develop malparesis in both hind limbs by 72 hours. So it actually gives us ideas that the locations of inoculations does not affect the clinical symptoms. So we look into details for the thalamus infected mice. It's split into two parts. One is within the brain itself. The second part is what we observe clinically. So within the brain itself, we're able to track the needle tracks because we are injecting with the 31 Gs of needle. And we actually found the virus is localized, allow the needle track at 24-hour post-infections. At 48-hour post-infections, the viral antigens at the injection sites, which means the needle tracks, is actually decreased. And at 72 hours, the injection sites, the needle tracks, the viral antigens was almost clear. So this shows that the virus actually have been cleared out around the needle track. Secondly, look at the clinical symptoms which compare with the peripheral organs. At as early as 24 hours, the brown fats, muscles have shown some infections, and it gets more severe throughout the time point. At 48 hours, we can find some of the neurons already in the spinal cord, which is a very classic way where the virus utilize the muscles motor neuron pathway at retrograde axonal transport and got into the spinal cord. Then it will spread to the brainstem. So this happens and found in 48 hours. And lastly, 72 hours, the severe distant bilateral infection in spinal cords and also anterior horns and also the brainstem. So these are taken from the figure ones of the papers. What we do is we get a cross sections of all the CNS regions. These are the spinal cords and these are the brain regions, and we track down of it. Just a brief review. So looking at the thalamus and hypothalamus regions, which is the second row, you can see the injected sites are heavily full with viral antigens at early time point, but really later clear off at the later time point, whereas the pons and medulla have severely infected at later time point. So in histo pictures, if you are interested, of course, you are welcome to get the paper and have a look. So briefly, 24 hours is here, 48 and 72, supported by H&E, ISA immunostains for the viral antigens, and ISH in situ hybridization stains for the viral RNA expressions. So all these have shown that at the injected sites, the viral antigens and viral RNA are both clear off at the later time point. So next we move to the second experiment, which we inject the virus directly into the pons and medulla, which is the hot area for infections in both mice and human. So similarly, at 24 hours, okay, the virus are highly localized at the medullary injected tracts. We found the needle tracts and is all over the place. At 48 hours, 72 hours, the local viral antigens decrease and the distant spinal cord anterior horns become heavily infected. So you will see some similarity between the pons inoculations and the thalamus inoculation mice. That is, the spinal cords, the times of the spinal cord get infected and the patterns of the CNS infections in later time point is both similar for both thalamus and medulla inoculation mice. So the temporal potence was nearly identical to the thalamus groups. So similarly, against because the needle tracks is quite near to the pons medulla, it's hard to differentiate that these come from the adjacent needle tracks or actually come from the retrograde axonal transport from the muscles, infected muscles. So we now we look at the spinal cords where the anterior horn cells are infected. So again, here are the H&E and the immunohistochemistry and in situ hybridizations that prove that the virus antigens are cleared off at a later time point. So besides histological analysis, we also did some of the viral titration study, both from thalamus inoculations and pons medulla inoculations. So the black and the stripes actually come from the pool muscles and the syrup, in the blood viremia. We can notice that the viremia is consistent throughout the experiments. Okay, around 3, virus titer 3. So this is also the same for the pons medulla. Whereas the pool muscle increased greatly, and we noticed that similar with our histo analysis, the virus actually presents quite a lot in the muscles. And looking at the thalamus inoculations, where the brain stems gradually increase, whereas the pons medulla, because these brain stems tissue are taken, not we just take the brain stem tissue for virus titration, but not the whole brain. So we can precisely track the virus quantity in the brain stem. So at 24 hours, this is due to the initial inoculant, and it drops and rises back at the later time point, which supporting our hypothesis. So now we are curious that whether these stereotypes or patterns or inflammations is actually related to the viral entry receptor, which is the SCARB2 receptors. So we plot and we analyze the expressions of the mouse SCARB2 in all over the brains and the other organs. In the thalamus and the pons, high expressions of mSCARB2, and we could not detect any SCARB2 expressions in the skeletal muscles and brown fats. Whereas we found differently in the expressions of EV-A71’s antigens. The antigens are restricted in the brain, which is very similar to our human autopsy cases. The infections of the presence of the CNS, the presence of the viral antigen in the CNS is actually very limited compared to other neurotropic viruses such as JE, where the viral antigens was actually massive in the brains itself. But EV-A71 is different. The virus antigens is very limited and is heavily surrounded by the inflammatory cells. So both in humans and mouse model. So we can see that there are differences in between the mSCARB2 expressions and the EV-A71’s antigen expressions. So that the mismatch actually suggested that the virus spread is highly restricted. It's not due to the, it could be due to the receptors expressions, but it also can due to other things. So in short, the mouse SCARB2 receptors actually does not play a very important role in the neuronal spreads inside the brain. So bypassing the receptor, we also use some of the primary and the, this is the neuroblastoma cell line. By direct infections, we notice that the virus actually is not very effective in the infections. Less than 10 cells are positive. But by doing the RNA transfections, it can give a robust replications around 30 to 50% are positive. So actually the cellular mechanisms can replicate the virus perfectly, but the barrier is at the viral entry that could not enter to the cells. So our hypotheses are once the virus are injected into the CNS, it leaks into the bloods and causing viremia, which leads to the massive muscle infections and lead to a retrograde axonal transports and re-enter to the brainstem and spinal cord. So these hypotheses have been supported by the evidence seen in the histopathological analysis and the viral titration analysis. So why is the local spread restricted? Why does the virus did not replicate in the CNS although we have injected directly into the brain itself? So we believe that the EV-71 actually having difficulties to enter the neurons where the dendritic processes of the cell body. So it prefer actually entry via the peripheral axonal terminal, okay, the neuromuscular junctions. And we also predict that the host immune response, which can be expressed by the neurons, for example, the AIM2 inflammasomes, the priortopsis, all these can cause the neuronal cell destructions and limit the replications of the virus, preventing the spreading effect. So in conclusions, we believe that the retrograde axonal transport is still the domain pathway for EV-71s to invade the CNS. And once inside the CNS, the EV-71's interneuronal spread is highly restricted. The mouse GAP2 does not dictate the viral tissue tropism in mice. So other receptor, for example, there are some of the supportive receptor, for example, the heparan sulfate, PSGL1s, may support the peripheral entry. So understanding this infection route is critical for developing a targeted therapy that blocks the axonal transports of the virus. So that's all for my sharing. So thank you. Any questions? So
Thank you, Dr. Tan. If you have any questions, let me just check the Q&A. Yes, we have questions from Prof. Kusara. So Dr. Tan, how are the viruses adapted for mice? And the second question is, what are the important clinical implications?
yeah, so the virus initially does not replicate well in the mice. So we injected the clinical isolated virus into the mice brains at the age of 1 day old mice. Then we collected the brains, we harvested the virus, and we injected into the new neonatal mice again. So we repeat the process for 3 times, and the newly isolated virus are now technically more suitable for the mice environment. So we injected it into the 2 weeks old mice, which mimicking appearance, and yeah, it caused a massive CNS infections in the mice. So that's how we get the mouse adapted viruses. So the next question is about the clinical implications. Yep, and so how does the virus get into the CNS is actually quite important for hand foot mouth disease or EV-71 viruses. So hand foot mouth disease actually caused by several virus, Coxsackie, EV-71s, but only EV-71 viruses actually can lead to the CNS infections, and is not a very common events. It's actually a rare event, probably 0.5% of the kids will get the infections. So in order to, we are trying to investigate why this kid gets the CNS infection besides his immune status or the body fitness of the current conditions. We also need to know how this virus get into the brain in order for us to develop the therapeutic targets and blockage of the virus entry into the CNS. And also, we do not know where is the primary replication sites in the human. is actually remain a mystery. We only know that the virus get into the body through the fecal oral mouth routes, and it get replicates in the hand. I mean the skin, the vesicle. You can see at the vesicles, sometimes in the oral mucosas, but how it gets into the brains are unknown. Yeah. So this help get us some of our ideas where to block them. Yeah. Thanks.
Okay, thank you. And we also have a question from Dr. Chan. How do you prevent the researcher from getting infected? Studies are done under level 3 standard safety protection.
Yep, this virus actually is a class 2 virus, so we've done it in the class 2 lab. Yeah, BSL2 lab.
And how do we protect the researchers?
So how to protect researcher? And actually, we don't have vaccines for this. The only safety way is this virus infects the children more frequently. It's quite a rare event in the adults. So we just treat it as the class 2 virus. We make sure that we only operate in the BSL's cabinets, BSL2 cabinets, and we will evacuate everything before we bring it out. So that's how general practice for the class 2 virus.
Thank you, Dr. Tan. And then we have one more question from Dr. Azlina. She's asking, at what time point after infection do the mice begin to show neurological symptoms? And do you see any viruses in the motor cortex?
Yep, and we did see some of the virus antigens in motor cortex at very late time points. For example, the 72 hours. And the viral antigens in the motor cortex is very rare and limited, probably just one or two. Okay, neurons are infected. Sorry, what's the question again? So, yeah,
So, yeah, do you see any viruses present in the cortex?
yeah. So, yeah, it's in the late time point. Just one or two neurons are get infected at 72 hours. So, yeah, when I see the CNS infections in the mice, yeah, from the clinical observations, we only noticed that at 48 hours, there are some high limb weakness, but we cannot be sure that is from the muscle infections or from the brain infections. From the viral titrations and the histological analysis, we did see the virus titers in the brain are present for 24 hours, and also the virus antigen presence in the pons and medulla at 24 hours. So the presence of virus in the CNS was observed at 24 hours at that um, concentrations of virus inoculated, but we do not see any symptoms observed that time.
Okay. Do we have any other final questions from the floor? I guess that's it for the morning. Thank you very much, Dr. Tan, for your presentation.
You
And thank you to all participants for joining us this morning.